by Finch Therapeutics for autism. Mark is the Chief Executive Officer and co-founder of Finch Therapeutics, which is a company developing novel microbiome drugs to serve patients with serious unmet needs. Mark has led much of the seminal work on engineering the human microbiome, publishing in leading journals such as Nature and New England Journal of Medicine. He translated microbiota transplantation from a topic of research into clinical practice by founding OpenBiome, where he pioneered the universal donor model as a safe, effective, and cost-efficient solution for the treatment of Clostridium difficile, the epidemic of Clostridium difficile infection. OpenBiome has now treated more than 55,000 patients through a network of over 1,200 hospitals. And Mark co-founded Finch to develop and scale a new generation of microbiome therapeutics. Mark graduated from Princeton University with a bachelor's in biology and received his PhD in microbiology from MIT. So without further ado, it's my pleasure to introduce Mark Smith.
Hey, John, thank you so much for having me here. I'm really excited to have the opportunity to share some updates on our work and a little bit more background on the microbiome and its potential linkages to autism. So I really appreciate the opportunity to share our work here today. So I will be making a few forward-looking statements as a publicly traded company. I need to share a few disclosures. Feel free to check out our website where we have a more fulsome set of disclosures available. So without further ado, I'll jump into the meat here. So as a little bit of context on the microbiome, we all have within us in our human genome about 25,000 genes. There are more than 20 million in our microbiome. So this is the community of tens of trillions of microbes that live on and inside of us. And this enormous genetic repertoire is now increasingly being understood through the application of high throughput sequencing techniques and analytical tools to really understand what this community is doing and the roles that it may have in our health. And what we've realized is that the microbiome has a really important role in modulating our immune system, our metabolism, and potentially even the function of our neurological system. And so at Finch, our objective is to really leverage some of these exciting tools from genomics and data science to come together to develop a sort of new class of therapies based on living microbial medicines. And so just a little bit of context on myself and my journey into this space. I actually started off as an academic researcher studying the human microbiome. My wife's cousin ended up with recurrent C. difficile about a decade ago, and it was a very, very challenging illness for him. He had been sick for about a year and a half. He had failed 18 rounds of, he had failed 7 rounds of vancomycin over that 18-month period. And, you know, for me, kind of watching his journey, I think there's some probably analogs to, what I understand a lot of families in this community feel, where there just really weren't good options available to him. He ended up sort of taking matters into his own hands, providing an at-home microbiota transplant, and it worked for him. But I just felt this is not a sustainable solution. This is not how we should be serving patients. And so I started an organization, as John mentioned, called OpenBiome to serve this community and make these kinds of treatments available for patients that had failed, all available therapies for treating recurrent C. difficile. I'm really proud of the impact that we've been able to have in the C. difficile patient population. And a little over half a decade ago, I started Finch with the objective of really scaling this sort of new platform of microbial medicines to other therapeutic areas and to develop a new generation of products that can really aim to build effective microbial therapies that can scale and really work for patients. And so a little bit about how we do that at Finch. You'll see if you jump on clinicaltrials.gov and actually just look up all the ongoing studies in microbiota transplantation. There's really been an explosion in research activity over the last decade, where now that you'll see more than 300 ongoing clinical trials exploring microbiota transplantation, a wide range of therapies. And our own view is not all of these are going to work. But those that do, deserve follow-up. And we have just an extraordinary opportunity to mine those clinical data to identify, what are the specific organisms and metabolic pathways that are actually driving the effects that we see in patients. And we think this is a really logical approach to address this sort of new therapeutic modality, where we can really learn from what's happening in the clinic, what patients are experiencing, and go through a process of what we call human first discovery. So reverse translation from what we're already seeing in the clinic to inform the kinds of therapies that we develop to scale in order to serve patients. And so we've been very successful in applying this approach to a number of therapeutic areas. So, we have a phase 3 program in recurrent C. difficile, having completed a positive placebo-controlled randomized phase 2 study just a couple of years ago. We have a great collaboration with Takeda focused on ulcerative colitis and Crohn's disease. And then the main focus of our time today, of course, will be on the work that we're doing in autism with FIN-211. And we also have a program focused on chronic hepatitis B infections as well. And so just a little bit about the tools that we've developed at Finch in order to address diseases that we believe may be linked to the microbiome. We have, on one hand, an approach that we call complete consortia. So all of us have living within us this community of tens of trillions of microbes. And what we're able to do is identify rigorously screened healthy donors and then harvest intact microbial communities from these donors, encapsulate them in enteric-coated capsules that we release at the appropriate location within a patient's gastrointestinal tract, and then establish a sort of new living community in these patients. And so on one hand, we have these complete consortia donor-derived products. And then we also have what we call our targeted consortia products. So where we've identified specific bacteria that we believe can have a big impact on patients, and then have isolated those, grown those up in pure culture, the way you might ferment beer or something like that, right? So these large anaerobic fermentation chambers. And then, again, encapsulated those in enteric-coated capsules that will release in the appropriate location in the GI tract. And what we're doing in autism with the FIN-211 program is actually combining both elements of this platform. So developing what we call an enriched consortia product, which has both a complete consortia scaffold, leveraging a lot of the exciting data that we've seen from microbiota transplantation, while also including an individual strain that's not found in all donors that we believe may be really important for serving these patients. And so we'll talk a little bit more about the FIN-211 program as we go, but that's the conceptual framework that we're using for the design and development of this product. And so for this community, I don't need to share how urgent the need is here. There's a really large unmet need. What I will highlight is, our development is specifically focused on the subset of children with autism that also suffer from significant GI symptoms. And we believe there are about a third of patients with autism that suffer from significant GI symptoms. We're specifically focused on those with significant constipation. And we believe that by focusing on this subset of patients, we can manage some of the heterogeneity that exists within the autism community and find a subgroup that we think is maybe more likely to respond to this therapy as an initial target for development, with opportunities to potentially expand the addressable population over time. And so, a little bit about what's driving our interest in this area and our decision to pursue a novel therapeutic based on the microbiome to serve the autism community. So, first, just a word on the epidemiology. We know that there are certain early childhood events that seem to correlate with the risk of developing autism later in life. So whether it's C-section birth or duration of breastfeeding or exposure to antibiotics, there are a number of elements that seem to be related to the composition of the microbiome that are epidemiologically associated with the risk of developing autism. And the antibiotics data, I think, is particularly interesting because we've seen a dose-dependent effect where individuals that have been exposed to more frequent doses of antibiotics during the first 2 years of life are at a significantly higher risk of developing autism later in life. So this kind of epidemiological framework is one clue to us that the microbiome may be one important element of the pathophysiology of autism. We've also spent a lot of time trying to understand why this might be the case. And one mechanism that we find interesting is the role of oxytocin signaling in autism. There's been a lot of work focused on delivering exogenous oxytocin, given the important roles that oxytocin plays in modulating sociality and bonding. What's particularly interesting to us about oxytocin is, it may be possible to induce endogenous production, so the production within an individual by modulating the microbiome, and we'll talk a little bit more about that. Another mechanism that we believe could be very important and could be relevant both for some of the behavioral as well as gastrointestinal symptoms that children with autism experience is the role of the gut barrier function in the etiology of autism. So we know that children with autism tend to have a less robust gut barrier. So they have sort of a leaky gut, which leads to the translocation of certain neuroactive microbial metabolites, like 4-EPS is one reasonably well-known example of these. And we know that microbial interventions can help to restore the integrity of the gut barrier. And this could be one way that we both improve some of the gastrointestinal symptoms and also avoid the circulation of some of these neuroactive microbial metabolites, and also potentially influence the neuroinflammation, which we believe could also be an important driver of some of the symptoms that are experienced by children with autism. And so I wouldn't, of course, be talking about any of this if it weren't for the clinical data that supports this. Again, our whole model for developing new therapies here at Finch is to really learn from what we're seeing in patients and in the clinic. And in this case, we're really excited that we've seen data from more than 200 patients that have been treated with microbiota transplantation across a range of studies. And each one of these studies has their own limitations that I think are important to highlight, and we'll discuss some of those as we dig into the data a little bit more. But taken as a whole, we find that this is really exciting proof of principle data that guides our development and really informs our understanding that the microbiome may play an important role in the development of some of the symptoms of autism. And what we've seen is, across these studies, we've seen improvements in both GI and behavioral endpoints, again, across a number of different investigational sites, different centers, really all around the world. And there's a recent Cell paper around the role of the microbiome and autism that highlighted, I think, the importance of this interventional data. The study was evaluating the correlations between the microbiome and autism, and found that they may actually be driven by an underlying correlation with diet in children with autism. And this is exactly why we've been so focused on interventional data. So looking at what happens when you sort of change the composition of an individual's microbiome and the impact that has on their symptoms and clinical outcomes. And this is why we believe that this reverse translation approach that we're using at Finch is such an important tool for really unpacking the role of the microbiome in this population and others as well. So now we'll dig into the data together. So I'll begin by walking through one of the first studies that came out in this space, run by Jim Adams at Arizona State University. And this was published in a series of papers starting in 2017. And in this study, it was a small open label study with the caveats that come with that methodology that conducted a single investigational center. And what they did is they provided a really robust series of microbiota transplantations over a period of an 8-week sort of induction process. And they evaluated these children both at baseline, 8 weeks after the cessation of therapy, and then even 2 years after these patients were last treated with a microbiota transplantation. And what they found was really encouraging. So in that 8-week time point, they already saw statistically significant improvements in GI symptoms. Those are preserved even 2 years off therapy. But the behavioral endpoints, I think, are the data that we found most encouraging. So, again, a statistically significant improvement at the 8-week time point. And at 2 years after treatment, a number of kids, sort of fell below what a threshold that has historically been used for identifying autism. And we found that these were really encouraging. Nonetheless, we've also said, well, this is an open label study, and I think there are some important caveats with that. And so, another data set that we've looked at to really inform our perspective on the potential role of microbial therapies in the treatment of autism is a study that used a comparator arm. So in this study, patients either received just behavioral therapy alone or behavioral therapy plus microbiota transplantation. And what we saw in this study is that these children again experienced significant improvements in GI symptoms and behavioral symptoms, and those were associated with changes in the microbiome, with increases in microbial diversity. And what's particularly interesting about this study and the next that I'll highlight is in these studies they found without the same really intensive induction therapy that was used in the Jim Adams study, they found that the effects seemed to wane with time. So there was an initial improvement, and then over time, some of the benefits seemed to wane. And this may inform the need for long-term maintenance therapy in order to maintain the benefits that are initially observed. And I think that's a really important question that we look forward to digging into in further detail. And the third study that I'll briefly highlight here, in part because it's just come out quite recently, just a few weeks ago, really builds on some of the other studies that I mentioned. In this study, children received either colonoscopically administered material or an oral therapy. And we thought this was important to evaluate the potential role of treatment modality. And when we found that there were no differences between those two modes of delivery, which is really encouraging for us because we believe that a colonoscopic intervention would be really challenging for children, but an oral therapy obviously has a lot of benefits in terms of access. And so these in this study, patients received a weekly dose of therapy for a 4-week induction period, and then were evaluated at 4 and 8 weeks after therapy. And so you can see that the GI symptoms improved quite significantly. And here we're showing the data around the Bristol stool scale, which is nice because it's a somewhat objective measure looking at the physical characteristics of stool samples that these children are passing. And at the end of therapy and even 8 weeks off therapy, there's some really significant improvements here in terms of the fraction of bowel movements that were abnormal according to the Bristol stool scale. Also really encouraged by the behavioral outcomes, and here we're using the CARS scores as in the earlier studies that I mentioned. And you can see the sort of temporal effects here, where there was a significant improvement at actually all of the time points, but that was most acute at the first time point, and there was perhaps some directional reversion that occurred in some of the later time points here. So, natural question again, getting back to some of the mechanistic concepts that I introduced earlier is, how does this work? And, I think there are a number of important caveats that are worth highlighting anytime we speak about, developing animal models of such a complex phenotype like autism. Nonetheless, I think these can be important tools to really try to understand mechanism because of the limitations of the kinds of work that we can do in clinical studies. And so in this animal model, where some of the key endpoints that were evaluated were the amount of time that these animals spent interacting with pairwise interactions with other mice, we found that interestingly, there's an individual strain that we knew was associated with induction of endogenous oxytocin, that when it was added, so the black here are neurotypical models, the red are the autism model, and then blue is the autism model treated with this oxytocin-inducing strain. And you can see that in this case we were able to restore neurotypical levels of these pairwise interactions by delivering this oxytocin-inducing strain. What's interesting is that was correlated with an improvement in restoration of neurotypical oxytocin levels. So, by itself, I think this would be difficult to draw any strong inferences from it. What's particularly helpful here is actually seeing some of the knockout work that was done as a follow-up to this. So if you vagotomize these mice and sever the vagal nerve, which connects the gastrointestinal tract to the central nervous system, you eliminate that rescue effect of adding in this oxytocin-inducing strain. So you can see that the interaction times are significantly lower among those mice that were treated with this oxytocin-inducing strain that had a severed vagal nerve. And sort of complementing that data, I found that if you knock out the oxytocin receptor, the gene that is responsible for the protein that binds oxytocin receptor, oxytocin, you can see that also eliminates the rescue effect of adding in this oxytocin-inducing strain. And so collectively, one of the inferences that we've drawn from these data is that oxytocin may be an important driver of in this model system, and that there are individual bacteria that we could deliver that are capable of restoring neurotypical behavior, and that was associated with increases in oxytocin, and it appears to be induced through a stimulation of the oxytocin receptor through a signal that is being transduced via the vagal nerve. So these are all we believe important early signals that may suggest a role for these oxytocin-inducing strains as part of a broader microbial therapy that could be used to try to replicate and build on some of the results that we've seen with whole community microbiota transplantation. And so this has heavily informed our product strategy here at Finch. We've chosen to deliver what we call an enriched consortia product, as I mentioned earlier. That's the design of FIN-211. And the idea is we have this complete consortia, which aims to really deliver an entire community, replicating some of the exciting data that we've seen with microbiota transplantation and this individual oxytocin-inducing strain, sort of the ability to deliver both elements of that. And we've had some really encouraging engagements with the FDA. In our pre-IND meeting, they agreed with us that we can go directly into children with autism. And in the Aspire trial that's going to be starting in the first half of next year, we're going to be able to do just that. We're going to be able to jump right in to start serving children with autism. And they also agreed with us that demonstrating benefit for either GI or behavioral symptoms could be sufficient to support a BLA. And we think that opens up a number of interesting development strategies for us in terms of how we may be able to advance this therapy to be available and accessible to patients in the future. So I'm very excited to introduce the Aspire trial here today. So this is a phase 1b study that's designed to really help us understand the optimal dosing level and dosing regimen that we can use as we carry this product forward into later stages of development. And so the part A, the first component of this study, is going to be a dose escalation study. So patients will either receive a low or a higher dose of FIN-211 for a 2-week period. And then we'll take the highest tolerated dose from the first phase of this study into a small randomized study where patients will get randomized to either receive antibiotic pretreatment, with the idea that by, similar to stem cell therapy, where you sort of want to knock out the incumbent community in order to create a blank canvas on which you can sort of paint this new ecosystem. Using antibiotics as a tool to standardize the baseline microbiome of children that are receiving this therapy, or patients with that don't receive antibiotic pretreatment. And our preference, if we can avoid the need for antibiotics, would be to move forward with a simpler therapy. We want to rigorously test that question in this first set of studies, that we have the right both dosing level and regimen, understanding whether we need antibiotics before we move into a larger placebo-controlled study. And the primary objectives will be looking at safety and tolerability, as well as the pharmacokinetics, so how these organisms are engrafting in patients. And we'll also be measuring as exploratory endpoints some of the key behavioral and GI endpoints that we discussed earlier. And so, really excited about the opportunity to begin evaluating these novel therapies in patients next year, and to start to get some early readouts from these trials in order to understand whether we're on the right track and we're going to be able to continue to advance these therapies. So I really appreciate the opportunity to come here again this year and share some updates on where we are. I hope that next year, when we present again, we'll have some early data from these studies. And really thankful to everyone in the community that's come together to help inform the design of this study. And I hope to continue to be able to work with the community to evaluate and understand and ultimately develop some really exciting novel therapies here. So thanks, and happy to take any questions.
Thanks, Mark. There's a few questions that have come in. So one, is the oxytocin producing strain named in the paper that you were citing?
Yes, it is. It's in the paper that's cited there. They use lactobacillus strain.
Great. Was that reuteri?
Yes, they used an L. reuteri strain in that study.
Great. Was that the same paper that showed a connection with BH4 metabolism as well?
It's the paper that I cited is a different paper, but by the same group. Okay.
One question for you in your trial. I know it's early days and maybe not a part of this study, but are you thinking about also doing any type of neuroimaging or anything of that sort to try and look at the actual, fMRI or some other sort of brain-based changes as a result of FMT treatment?
Yeah, it's a topic that we spent a lot of time discussing with the team, and kind of weighing some of the pros and cons. We want to minimize the burden on kids and their families, but we're also really interested in that data. And so I don't know that we've made a final decision on whether we're going to include those in at some point in the study, potentially as a sub-study. But the expectation is that won't be a requirement for the study at this point, but it's a topic that we're continuing to discuss.
Cool. One thing that might be interesting, if the microbiome, oxytocin, BH4 side of the story pans out in humans, Richard would be a good person to talk to. One of the studies that we published at Arkansas Children's was actually on tetrahydrobiopterin or BH4 treatment, Kuvan for autism. We showed that there appeared to be a connection with nitric oxide metabolism, and we theorized that maybe it had something to do with improving cerebral blood flow. So be curious if FMT treatment may actually have effects on nitric oxide metabolism, and if that actually could be shown up if you do decide to do any fMRI type studies and see changes in cerebral blood flow patterns. Might be neat. So a couple other questions have come through. If the study shows that vancomycin pretreatment is important for effectiveness, is it possible to extend the same to placebo group as well, since they may be left out of the FMT treatment protocol?
Yeah, it's a great question. So in this study, Aspire Part B, there won't be a placebo group. So actually everybody's gonna have access to therapy. The group that's only receiving FIN-211 won't get the vancomycin therapy as part of it, because there could be efficacy just from the vancomycin component, which is one of the questions that we want to understand as part of the study. But there won't be a vancomycin standalone arm in this study.
Okay. Another question that's come through, and I think this will be the last question before we move on to the next presenters. Do you have a theory or any rationale for why there might be a need for multiple FMTs for children with ASD? Following to that question, maybe an underlying mechanism affecting the microbiome that requires certain bacteria to be constantly replenished. Have you thought about that? And do you have any suggestions there?
Yeah, so it's a great and actually very complex series of questions around this sort of ecological dynamics and sort of longevity of these communities. The short answer is, children with autism, they have if there are sort of disruptions to their microbiome, it may be driven by, dietary preferences or other, elements that they experience. And so some, whether it's, genetics or their environment or diet, something has about those confluence of factors have selected for a dysbiotic microbiome. And so, part of our thinking is that without constantly sort of replenishing this, some of those missing microbes, they may be lost over time as they have been potentially by some of those factors. And so that could be one reason. And it's something we're really excited to be able to study here, is we'll be able to actually measure how the community changes over time and actually use the sort of community decay in part as, to inform our understanding of how long dosing may be necessary for in the future. So that's how we think about engraftment and pharmacokinetics in this context. And it's a really important and complex part of how we think about developing these kinds of therapies in autism and in a range of other applications as well.
Great. Then one last quick question, whether it be related, just in general, it could be related to autism or just, in general in terms of FMT treatment. Curious, do you know of any reports on really any condition of doing a microbiome transplant therapy and that affecting dietary preferences after the fact?
I don't know of any rigorous studies of the question, though anecdotally, I've heard of some pretty significant changes in dietary preferences among C. diff patients, though those are kind of to be expected because they're, have very, very severe diarrhea and are, often experience significant weight loss and then, have their appetites restored once the probably the underlying driver of that is resolution of their C. diff and the underlying diarrhea. But in a kind of more normal population, I haven't heard examples of that yet.
Great. All right, Mark. Well, it was always a pleasure. Thanks for joining us. And there might be some more questions. If you wouldn't mind looking at the Q&A and the chat function and get back to folks, I'd be greatly appreciated.
Yeah, absolutely. I'll do that offline right now and let you guys